Now that we have looked at the structure of DNA, we want to look at DNA replication. How does DNA copy itself? There's three models that were originally presented for the way that DNA could replicate itself. These three suggested models are pictured here on the right. In this diagram, the gray strand represents the parental strand, or the original strand, and then the blue is a newly built strand. So anything that is built with new nucleotides is blue, and anything that was an existing DNA nucleotide or strand is in gray. There's three possible ways. One is conservative replication. When you think of conservative, think of this is your original DNA molecule. And when you're done with replicating, you still have your original DNA molecule that hasn't changed. So it has been conserved. And then you have an entirely new DNA molecule. The original double strand stays intact, and you completely synthesize two new strands in your new DNA molecule. Next, we have semi-conservative. If we look here, we will see that this original DNA strand is not completely conserved, but partially conserved. So in our two new DNA molecules, one of the strands is the same as the original strand, but then the other side of the DNA molecule is new. That is semi-conservative. In this case, the two strands will come apart, and we're going to use each of those strands as a template to build the new strand. In dispersive replication, there's no conservation of the parental strands. We don't see an original parental strand anywhere here. Essentially, what we have done is we've chopped up both strands of the DNA and used new nucleotides and interspersed those new nucleotides with the old nucleotides to get dispersive replication. Both of the original parental strands are broken up into pieces and then resynthesized. Now we want to look at an experiment that determined which one of these models was accurate. These experiments were done by Michelson and Stahl. They used bacteria, and they also used isotopes. They used an isotope of nitrogen that is a heavy nitrogen, so it has a molecular weight of 15 instead of 14. They took this DNA that they had synthesized in heavy nitrogen so that all of the bases in that DNA molecule would have this isotope that is heavier than regular nitrogen. And that's what we are seeing here in this red. So that is based on growing this chain in heavy nitrogen. So you start off with a DNA molecule where all the bases have this isotope. If you take your DNA, you put it into a solution of cesium chloride and centrifuge it, it'll form sediment. And the sediment will appear at different places depending on the density of the DNA molecule. So as we would expect, if I was to look at this in a tube, in a centrifuge tube, the heavy band with the heavy nitrogen should be lower in my tube, and then the lighter band that only has nitrogen 14 in it should be higher on my centrifuge tube. If I had some that was just purely N14, it would all be up here. They took their initial heavy DNA molecule and put it into a media where the only bases available to it were nitrogen 14. So we no longer have heavy nitrogen available to it. And now we allow that original DNA molecule to replicate. This is what we see when we first start, before any replication. After one round of replication, the band forms here. We only get a single band, and it's a little bit lighter than what the heavy nitrogen would be, but it's also a little heavier than what nitrogen 14 would be. What this is telling us is that all the molecules that were in here are essentially in between heavy nitrogen and regular nitrogen, telling us that each one of those DNA molecules contains 50% heavy nitrogen and 50% N14. That's what we're seeing here. So we started off with both strands being heavy. Now at the first generation, one strand is heavy and one strand is light. So they're intermediate where they come out in the centrifuge tube, but they're all the same because they both have one strand that's heavy and one strand that's not heavy, and that's why they're at one particular location there. Now we let them go through again, but once again, only N14, no more N15 in here. Now what's going to happen? This strand right here will build a new strand off of it with the regular N14. That's that right there. This strand here on the right of this first one will build a new strand with N14, but now both strands have N14. The same thing is happening with this one. Both strands are N14. Here we have N15. Its new strand is N14. Now half of my DNA DNA sample is going to be here at a combination of light and heavy nitrogen. And then 50% are going to be up here higher because they only have N14. If we continue that same logic, so once again, we're going to go through replication. This one will create one heavy and one light chain. This one will be both light. This one, of course, will do all lights. And then here at the very end, we'll have one that has a heavy and a light. Now, if I look at all my different DNA molecules that are in there, in generation 3, 25% of them are going to be a mixture of heavy and light, and 75% of them are going to be all light. As we continue to go through generations, we are going to increase the percentage of this light band here and decrease the percentage of the ones that's the mixture of the heavy and light. In later cell divisions, we have an increasing amount of DNA that only has N14 in it, no N15. You can see, whereas if this had been dispersive, for instance, all of those molecules should have had some N14 and some N15 in them, but they didn't. The data support the semi-conservative replication model. Now let's take a look at some of the specifics of DNA replication. First, we'll look at prokaryotes because it is slightly different for prokaryotes and eukaryotes. It's much more understood in prokaryotes because they are much simpler systems than eukaryotic cells. A typical prokaryotic chromosome is a circle, and so first we have to find a place to start our replication. So there will be a particular sequence on the DNA that is recognized as the origin sequence. We see this here. There's a certain sequence, this ORI sequence, that is recognized. That's where a particular protein complex will come and bind with that and start to open that molecule up so we can start to unzip it. Helicase is an enzyme that's going to separate those strands, and we can see them separating here. Now, when they separate, they form what is known as a bubble right here, and on either end of the bubble, this is referred to as a fork. We have a bubble with two forks on either side. As we open it up, this fork is opening in this direction, and this fork is opening in that direction. Down here, we can see this fork and this bubble in a little bit more detail. We see here this helicase enzyme. That helicase is opening this up, and it's doing it on both ends, so it's going in both directions and unwinding it. We form that replication fork. There's actually two forks, as we said, on both sides. So here's our bubble with a fork on either end, so each bubble has two forks. Imagine taking a twisted piece of yarn and opening it up like this. You know what's going to happen is the two ends on both sides here are going to start to knot up. As you pull this open, you're going to get knots forming here. This enzyme, topoisomerase, its job is to prevent those knots from forming. As it opens up, it will prevent it from getting knotted up. It can make nicks in the molecule and put it back together to relieve the strain on that molecule so that we don't get that overwinding and that stress on the molecule. So here we see topoisomerase keeping this from getting tangled. DNA has an inherent characteristic, and that is it wants to go back together. This molecule really likes to form the hydrogen bonds that it had with its bases before, and it will naturally do that. And so what we also have to have is a protein that prevents that helix from reforming, and this single strand binding protein does that. We see the single strand binding protein here, keeping that replication bubble open while we are copying it. These are some of the enzymes and proteins we have working right now. We have helicase opening it up, topoisomerase preventing it from overwinding and getting knotted up, and then the single-stranded binding protein keeping it from forming that helix again and coming back together. After we've opened it up, we're going to look at what's going on one of our forks. So we're focusing now on this particular fork. We're going this direction. We know that this is also happening on the other side of the bubble at the other fork. First, we need some RNA primers. There's an enzyme called primase that synthesizes these RNA primers. And the reason we need RNA primers is you can't build a DNA molecule without a starting point. We have to have something to start with. So if I'm going to bring in one of my nucleotide building blocks, I have to hook it to something. That's what the primer is doing. This primer is giving us a place to hook our nucleotides to when we bring them in. We have to have this starting point to attach them to. RNA primase enzyme helps us build this primer. Now, one of our strands has one primer for this particular fork. Ignore the other fork for now. For this fork, we have one primer up here, Now down here on this strand, we have many primers. This one is showing 2. When we get ready to add our nucleotides, this is our nucleotide, DNA polymerase III in prokaryotic cells is the enzyme we're going to use to start adding these sections. And we're going to first add it to a primer. So we have this primer, and now we're going to start bringing in each of those individual bases to match the one that's on the other side. This red base will match whatever its partner is over on the blue side. So if I have A on the blue side, I'm going to have T over here on the red side. I'm going to bring in the appropriate matching base. And I'm going to hook it to this primer. Think of it like a pop bead. I'm going to bring it in, I'm going to pop it together like a Lego or something. Once I have my primer, I can start bringing in those nucleotides and building my new strand. Here is something that's really important to remember. This polymerase III can only add nucleotides to the 3' side. It must add down here to this OH group. I cannot add a nucleotide up here. I have to add it down here. And that will be important because down here I have a 3'. And so if I'm building this molecule, I can add them here. If I'm building this molecule and I'm trying to go this direction, I can't because I can't add my next nucleotide here because I don't have a 3' side. I can add it up here, but I can't add it down here. Let's look at some of the other steps involved. Since polymerase can only add it to the 3' side, one of my strands can be built continuously. The other strand has to be built in little pieces, in little fragments that I then later stitch together. As we saw a minute ago, this one can build in this direction because I can add to my 3' side and bring in new nucleotides going this way. But I can't do that over here. On this side, I have to build it the other direction. Since our two strands are going to be built differently, we refer to them as our leading strand and our lagging strand. Our lagging strand has to be built in pieces called Okazaki fragments, and our leading strand can be built continuously. Let's take a look at this for now. We are opening this molecule up going to the right. I have my anti-parallel strands, so this is my original template strand. Here's my template strand I'm using to build from. It's running 3' to 5'. I know that the strand I'm going to build off of it has to be in the opposite orientation. So for this green molecule on the top, my 5 is here on the left. This end over here is my 3' end. As helicase opens this up and unzips it, what is exposed right there is 3'. So we're in good shape. It exposes a 3'. I can bring in a new nucleotide base using my polymerase III and attach it, and everything is fine. That is called my leading strand. My leading strand I can continuously replicate. I just keep opening it up and it just keeps going down. My other strand is the opposite orientation. My template down here on the bottom, this is 5' on the left and it's going to 3' on the right. The one I'm building off of it is 3' on the left, 5' on the right. When I open this up, what do I see? I see 5'. What can I do with that? Nothing, because I can't add to 5'. What we do on this side is we open it up a little bit further, jump ahead, we add in a little primer, and if I put in a primer, let's say right here, and now that primer is 5' up here on the right and 3' right there, now I can bring in bases and build it in backwards. And then I can open this up a little bit further, jump ahead, build it backwards. Open it up, jump ahead, put in a primer, build it backwards. That's what we're going to do on the lagging strand. So on this fork going to the right, this top strand is my leading strand and the bottom one is my lagging strand. If I was looking at this same thing but I was going the other direction, then the leading and lagging are going to switch because I'm going the opposite direction. Another thing that has to happen is later I have to go in and remove those primers because I have these little pieces that are up here that they're not really showing here, but I have little pieces here that are these primers that I have to get rid of. So I have an enzyme for that as well. DNA polymerase I replaces that RNA primer with DNA, so it can come in and pop in those extra DNA nucleotides that we need. After we do that, we have to take these Okazaki fragments and hook them together because we're going to have a bunch of pieces here. And to do that, we use DNA ligase. Let's take a look at this in more detail in a diagram. Here we have this replication bubble. We're going to look at this fork going towards the right. So we're synthesizing this going from left to right. We have used helicase to unzip the molecule. We're using this topoisomerase right here to keep it from tangling in front of where we're opening it. If we look here at the bottom, this is our 3' end on this side. This is 3 here and it's 5 over on this side. So what is this one? This is 5 on the left. So as this opens up, what am I seeing? I'm seeing 3. This DNA polymerase associates with some other proteins. They call that the sliding clamp, and it moves down. Every time we open up, we can just add new bases in. And this single strand binding protein is keeping it from reforming here as well. This is our leading strand. We're continuously replicating it. No big deal. Over here on our other side, this is 3' on the left, but it's opening in this direction. When this opens up, what I see is a 5' end. I can't add to a 5' end if I'm DNA polymerase. Primase comes in, puts in a little RNA primer. Now over here is 3'. Now I can build some DNA off of that. So here's a primer. I built my DNA backwards. Here's a primer. I'm going to build DNA in here. I can put in a spacer that shows me the 3' end of RNA, and then I can bring my bases in, attach them to that 3' end, and then later I'm going to come and remove those primers using DNA polymerase 1, and then hook my DNA sections back together again using ligase. This video will animate this for us.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
One thing I want to point out here as well, because you'll hear it stated this way a lot, we said that DNA polymerase III can only add the 3' side. That means that if I look at a growing strand, it is growing from the 5' to the 3'. The direction of growth is going this way. And the same thing is true up here, because up here, this is the 5' side and that's the 3' side. So if I look at the way my DNA strand is growing, it's from 5 to 3. So we talk about DNA being synthesized in a 5' to 3' direction because we're adding on to this 3' side. So I want to make sure that that's clear. The other thing that was mentioned in the video that we don't have here is it talked about the primers being taken out by the exonuclease, which is an enzyme that removes bases, and then filled back in using polymerase I. This gives us a summary of some of the enzymes and proteins that were important. Polymerase I, which is the one that replaces the primer. Polymerase III is the one that is the main enzyme that adds those nucleotides in that 5' to 3' direction. Helicase is the enzyme that opens up the DNA, unwinds it. Ligase is the one that comes and seals those Okazaki fragments back together. Primase was the enzyme that inserts the primers. The siding clamp is proteins that hold that DNA polymerase in place as it moves down the DNA molecule. Topoisomerase was the one that keeps it from tangling. As it's unwinding, it breaks and then reforms it so that it doesn't get all tangled up. And then the single-stranded binding protein were the ones that kept it so that the DNA helix didn't reform. Keeps those two strands single. Let's briefly look at DNA replication in eukaryotes. The steps are essentially the same as they are in prokaryotes. It's just that because we have more complex genomes, larger genomes, we don't have a circular chromosome, we have many linear chromosomes, it's going to be a little bit different. We also have multiple origins of replication. If you remember from our prokaryote, we started at that ORI sequence and we moved in both directions around the circle until we got to the other side. Here, we are going to take that linear chromosome and we're going to open it up in lots of different places, not just a single origin. We can see that here, where along the length of this chromosome, we have many different replication bubbles, lots of different replication forks, and they are going to proceed towards one another. And we can also see that down here. Even with all of these multiple origins, the rate of replication in eukaryotic cells is still a lot slower than it is in eukaryotic cells. There's a lot more DNA material to copy. There's also a lot more polymerase molecules. So we know that there's 14. 5 of those have been well studied. Several of the ones that have been studied pretty well. Polymerase alpha is the one that complexes with primase to make that first primer. So polymerase alpha is somewhat similar to polymerase I that we saw in prokaryotic cells. Polymerase delta and epsilon add bases. Remember that these eukaryotic chromosomes are also bound to histones and form those nucleosomes. And so in the process of replication, those histones have to be removed and then put back on as well. Here we'll watch another video on replication.
Not only know the basics of DNA structure, we need to understand how it copies itself because cells are constantly dividing, and that requires a complete copy of all that DNA information. It turns out that our cells are extremely good at this. Our cells can create the equivalent of 10,000 copies of this book in just a few hours. That, my friends, is called replication. Every cell in your body has a copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. So, how is a teenage boy like the enzyme helicase? They both want to unzip your genes. Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand as I call it, and another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt to deal with. These unwound sections can now be used as templates to create two complementary DNA strands, but remember, the two strands go in opposite directions in terms of their chemical structure, which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading, good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it could do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning, then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging or scumbag strand is, well, he's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5' 3' direction, and the lagging strand is 3' 5'. So DNA polymerase can only add new nucleotides to the free 3' end of a primer, so maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments, and here that awesome little enzyme RNA primase does its strands in the 5' 3' direction, and the lagging strand is 3' 5'. So DNA polymerase can only add new nucleotides to the free 3' end of a primer, so maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments, and here that awesome little enzyme RNA primase does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments, after the couple of married scientists who discovered this step in the process in the 1960s. And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki-someone's-someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers, and then the little fragments gets joined up by a final enzyme called DNA ligase. And that is why I say that the lagging strand is such a scumbag. DNA replication gets it wrong about 1 in every 10 billion nucleotides. But don't think your body doesn't have an app for that. It turns out that DNA polymerases can also proofread, in a sense removing nucleotides from the end of a strand whenever they discover a mismatched base. Because the last thing we want is an A when it would have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. Remember, we're talking about millions of miles worth of this stuff inside us, and this, my friends, is why scientists are not exaggerating when they call DNA the most celebrated molecule of all time. So you might as well look this episode over a couple of times and appreciate it for yourself. And in the meantime, gear up for next week when we're going to talk about how those 6 feet of kick-ass actually make you you.
Now we want to talk about the replication of telomeres in eukaryotes. Telomeres are the ends of the chromosomes. So clearly this is not an issue in prokaryotic cells that have circular chromosomes. But when you have linear chromosomes, telomere replication can be an issue. Remember that when we go to replicate the lagging strand, we have to jump ahead, put in a primer, and build those pieces backwards. How do you jump ahead when you're at the end of the chromosome? There's nowhere to jump ahead to insert that primer. So we have an issue replicating the telomeres of these linear chromosomes in eukaryotic cells. Where do you put the primer? There's no way to jump ahead and put that primer in on that lagging strand. Potentially, every time that chromosome replicates, it could get on that one strand shorter and shorter because of the loss of the telomere. There's an enzyme called telomerase that can add those telomeres back in. So as we see here, telomerase would come in and add a section so that we could continue to make that primer and synthesize the rest of that molecule. Here we'll see a few videos on telomere replication.
What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. They cap and protect the end of a chromosome like the end of a shoelace. In almost all animals, from the simplest to the most complex, telomeres are required for cell division. With each cell replication, the telomeres get shorter and shorter until they're so short that your cells can no longer divide. When cells no longer divide, tissues age. However, telomeres can be rebuilt by an enzyme called telomerase to restore cell division. If you think we've found the fountain of youth, think again. If a cell keeps dividing uncontrollably and overcomes the limitations of telomeres, a cancerous tumor can form. This is why telomeres are so important. In healthy cells, they achieve the necessary balance between limiting cellular lifespan and your own longevity and keeping cancer growth at bay.
The ends of linear chromosomes pose unique problems during DNA replication. Because DNA polymerases can only elongate from a free 3' hydroxyl group, the replication machinery builds the lagging strand by a backstitching mechanism. RNA primers provide 3' hydroxyl groups at regular intervals along the lagging strand template. Whereas the leading strand elongates continuously in the 5' to 3' direction all the way to the end of the template, the lagging strand stops short of the end. Even if a final RNA primer were built at the very end of the chromosome, the lagging strand still would not be complete. The final primer would provide a 3' hydroxyl group to synthesize DNA, but the primers would later need to be removed. The 3' hydroxyl groups on adjacent DNA fragments provide starting places for replacing the RNA with DNA. However, at the end of the chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosomes would progressively shorten during each replication cycle. This end replication problem is solved by the enzyme telomerase. The ends of chromosomes contain a G-rich series of repeats called a telomere. Telomerase recognizes the tip of an existing repeat sequence. Using an RNA template within the enzyme, telomerase elongates the parental strand in the 5' to 3' direction and adds additional repeats as it moves down the parental strand. The lagging strand is then completed by DNA polymerase alpha, which carries a DNA primase as one of its subunits. In this way, the original information at the ends of linear chromosomes is completely copied in the new DNA.
Telomeres seem to be implicated in aging and cancer. So only germ cells, which are producing gametes, and stem cells, which are cells that can give rise to other types of cells. In stem cells and germ cells, telomerase is expressed. That gene for making telomerase enzyme is active and the enzyme is being expressed. Elizabeth Blackburn, Carol Greeter, and Jack Szostak received the Nobel Prize for the discovery of telomerase and how it works. Somatic cells that do not express telomerase lose their telomeres as they divide. So these get shorter and shorter as they divide if they don't have telomerase being expressed. There is some connection, although it's not 100%, but there is some connection that aging might be in part due to the loss of telomeres. Or put it this way, they seem to be correlated in some cases. As aging continues, telomere loss is also occurring. Organisms that age faster in some studies maybe have shorter telomeres. The data are mixed. In some cases, they found no correlation there. In other cases, there has been a correlation found. It's still up for debate. But you can see here that we have a chromosome for the adult cell here. After many replications, then this telomere can be shorter and shorter and shorter. As the cell ages, these telomeres get shorter. If we have telomerase, then those telomeres do not get shorter. So this would be essentially an immortal cell. If any of you have read the book, The Immortal Life of Henrietta Lacks, they took cells from this woman back in the 50s, and these cells are still being used for most of research and medical technology today because that cell line is still continuing. And these cells just never stop dividing, where most cells do. They have shown then that in many cases, this telomerase activity is associated with that. It also seems to suggest that cancer cells have shorter telomeres, and the telomerase is more active. What a more active telomerase means is that these cells could undergo more replications, which of course is not what we would want in a cancerous cell. We would like that perhaps to be the case for a healthy cell. If we could target cancer cells and inhibit their ability to express telomerase, we might be able to get those cells to sort of age and die more naturally so that they don't continue to reproduce. In the same token, we might be able to take healthy cells and express telomerase in them so that they can survive a little bit longer. Obviously, we have to be careful with that. We don't want to take a cell and turn the gene for telomerase on and then have that cell go rogue and it never stops dividing. So obviously, there has to be some control over how we manipulate this. There's an article here that talks a little bit about how telomere length can be used as a marker of biological age. Here, watch a video on telomeres aging in cancer.
I hate death. More than 150,000 people die on this planet every friggin' day, and some of those people are getting hit by buses or dying of how cute a puppy is or something, but in most cases, their bodies are just succumbing to the dirty work of aging. And I'm tired of it. And I don't want George R.R. Martin to die before he finishes a Game of Thrones series. Obviously, we all have to die eventually. I mean, it's something that we have in common with every other person who has ever lived, but some organisms can live to be very, very, very old, and scientists are beginning to understand better how they do it and what it might take for us to live to be very, very, very old. And I'm not just talking about doing squat thrusts or eating kale or whatever, glass of white wine every day, although those things probably help. I'm talking about discovering how our cells die, why they do it, how our genes make us age, and yes, how our behavior influences our longevity. So come on, scientists, make me live forever, please. You and I are just collections of cells, albeit magnificent and really complicated collections that exist as animated blobs for a while, hopefully long enough to reproduce. And when it gets too tough for our cells to keep these masses of cells together, we die. It happens to the best of us. And also the worst of us, and also everybody in between. For humans, and for most animals, there's a process of aging that happens over the course of our lives, culminating in the buying of the proverbial farm. This process has a lovely little name. It's called senescence. And at an organismal level, we see senescence take place after our bodies reach sexual maturity. After that, we begin to lose the ability to combat physical stress and to maintain homeostasis, the internal balance of all of our organ systems and body fluid concentrations. And most importantly, we start to lose the ability to combat disease. Because technically there is no such thing as dying of old age. Everybody dies of something. But this is good news. Not all animals do this. Not all animals age this way. Some, particularly some cold-blooded animals, exhibit what's called negligible senescence. They don't lose their ability to reproduce over time, and their death rates don't necessarily increase with age. These animals basically stay at a certain level of fitness until something terrible happens to them, like a disease or predation or a really big, fast truck barreling down the road that they're about to cross. So, for instance, Galapagos tortoises have been known to live for 170 years, and lobsters can live to be 140, and the oldest quahog clam ever collected was about 405 years old. Frickin' Shakespeare was writing Hamlet when that clam was born. So that's totally unfair. Why do some animals get to start to die as soon as they reproduce, but others get a hall pass to near immortality? Scientists don't know for sure. They're trying to figure it out by studying senescence at a cellular level. And it turns out that our stupid cells have their own deaths programmed into them. Our somatic cells, the cells that aren't sperm or eggs, are constantly dividing and making copies of themselves. In fact, there are whole armies of cells being made in your body right now. In the 1960s, a young researcher named Leonard Hayflick was studying human fetal cells when he noticed that after a while, human cells just stopped dividing and then they died. He came to realize that they all quit dividing after about 50 divisions, which took around 9 months. If he put healthy dividing cells into the freezer, the division would slow down and even stop, but when he warmed them back up, they'd remember exactly where they left off and start dividing again until they got to that magic number. It was in this way that Hayflick discovered that human cells have death programmed into them. The number of times a cell can divide is now called the Hayflick limit, and although it's 50 divisions for human fetal cells grown in a culture, the number for some animals is less and for others it's more. For instance, mice, which can live for 2 or 3 years, go through anywhere from 14 to 28 divisions. A Galapagos tortoise, on the other hand, has a Hayflick limit of about 125. So there may be a correlation between an animal's average Hayflick limit and its lifespan, but as with everything in science, it is not this simple. Because even though we're always making more new duplicate cells, as we get older, that Hayflick limit gets older. Studies of people in their 80s and 90s found that their cells only divided about 20 times. So since our cells have expiration dates, we in turn also have expiration dates. By now, good scientists are asking, okay, yes, there's a limit, but what causes the limit? Turns out the answer is probably its chromosomes. A human cell quits dividing after it stops being able to completely replicate its telomeres, the little caps of non-coding DNA on the end of each chromosome that protect the genes from errors in copying. Telomeres are originally made with the help of the enzyme telomerase when we're just little zygotes. But after that, every time a cell divides, the telomeres on the chromosomes of a new cell are a tiny, tiny bit shorter than those in the parent cell. A cell reaches its Hayflick limit when the telomeres become so short that they can no longer protect the chromosomes, and the cell becomes unviable. So, why don't biologists just fiddle around with our cells and add some telomerase to the mix so that the cells can keep dividing forever? Well, there's actually a kind of cell that already does that really well. Cancer cells. Yes, cancer cells can sometimes create their own telomerase so that they can replicate indefinitely without their chromosomes getting damaged, which is why they divide like crazy and become tumors. Because of that, nobody's super anxious to start injecting people with telomerase. And in fact, one theory is that programmed cell death evolved in order to keep our tissues from undergoing explosive cancerous growth. So, senescence, one thing that we have to grapple with as mere mortals. But another thing that's keeping us from playing basketball until we're 500 is our genes. Here, research into extending longevity has focused on a little nematode called C. elegans. These little worms are really good test subjects because their lifespan's only about 14 days and they only have about 20,000 genes, so scientists have been able to pinpoint the genes that are most likely in charge of aging. In 1993, Cynthia Kenyon, a biologist at the University of California in San Francisco, found that there was just one gene that was making these worms age. It's called DAF2, and when she messed around with that gene, basically mutated it so that it didn't work, her worms started to live 28 days instead of just 14. Twice as long. If they did that for us, we would live to be like 160. And not only that, the worms were like spunky and vivacious all the way until they croaked. Kenyon also found another gene called DAF16 that has the opposite role. It keeps the worms young and healthy by regulating the production of antioxidants, germ-fighting proteins, and other things that fight off pathogens and the effects of stress. Because, you know, being a worm, stressful. So what Kenyon found is that the aging gene worked by restricting the effects of the longevity gene. And when you damage the DAF2, DAF16 just keeps on doing its business, keeping the worms young. Now, that's all well and good for the worms. I'm glad that they're having nice, long, healthy lives, but I'm not really too concerned about them. What about us? Well, the mammal scientists have indeed found two counterparts to that nematode aging gene, and in humans, a lot of research has focused on one gene in particular that creates a growth hormone called IGF-1. When scientists silence this gene in mice, there is less cell and organ damage caused by oxidation. Organs seem to be less susceptible to cancer and other age-related illnesses, extending the mice's lifespan by up to 33%. Now, why aren't we injecting George Martin with this stuff already? Well, for now, scientists aren't ready to start exploding everybody's genes. Just because it works in mice doesn't mean it's going to work for people. But it does lead us to one more final cause of aging that scientists are investigating: calories. That's right. It turns out that even though we need calories to live and work, the calories we take in are also stimulating the aging process. Scientists have known, actually since the 1940s, that lower caloric intake leads to longer lifespans in mice and other animals, but it wasn't until recently that they put their fingers on the possible cause: that calorie intake stimulates IGF-1. How that works isn't exactly clear. In fact, it's one of the bigger mysteries in the study of aging. But one theory is that since IGF-1's ultimate job is to direct food energy toward growth, if you take in fewer calories, your metabolism shifts gears from growing to simply maintaining existing cells and bolstering your resistance to stress and disease. So getting bigger and stronger becomes less less important than just staying alive. But before you put yourself on an all celery and watercress diet, don't, because again, just because something increases the lifespan for a mouse doesn't mean it's going to have the same effect on you. Are you a mouse? If you're a mouse, then do that. Scientists have been doing calorie intake studies on animals more like us, monkeys for instance, but it's tough to do longevity studies on long-lived animals because they take freaking forever because they're like not dying. The longest running human longevity study has been going since 1921, which is pretty cool. Good job doing science back in the day when our subjects were about 10 years old. Some of the kids involved in that research are indeed still kicking. Good work, folks. But as you might expect, many of the researchers have since died as well. One of the pitfalls of studying aging. So it might be a while before somebody's willing to tinker with your cell chemistry or genetic code to help you stay young and spunky forever. But in the meantime, you can take a look at your family. How old are they living to be? How long your grandparents and parents live probably accounts for about 20 to 30% of your chance of living past the ripe old age of 85. Otherwise, lifestyle choices like diet and exercise and whether or not you smoke play a pretty obvious role in longevity. Studies show that Seventh-day Adventists, always great research subjects because they're an actual religion that encourages regular exercise, vegetarianism, and refraining from smoking and alcohol, have an average lifespan of about 88 years, about 8 years longer than the average US citizen. So while our best and brightest keep researching senescence, genetics, and calorie intake, just don't smoke like Kate Moss, eat like a pig, or drink like a fish, and you may live longer than those things at least.